Hi folks, welcome back. Today I want to discuss what we learned from the process of replication. And in the previous video, I explained that the process of replication is how the DNA molecule is copied. And as I explained, it's a complicated and amazing process. Each strand of the DNA is copied in a different direction, each one using different mechanisms, yet still using the same enzyme, DNA polymerase. So we have two different processes in two different directions, yet only one enzyme, copying the DNA in both of these processes and direction. So my hope is that you can understand how absolutely amazing that is. And yet again, as I've explained in previous videos, we have more patterns all over the place in replication. And therefore, as I've explained before, we have another element of design. We see a pattern in the processes and directions of replication used on each DNA strand, what we call the leading strand and the lagging strand. This tells us that both intelligence and prescience are an absolute requirement for and during the creation of the process of replication. You must not only know the two different processes needed for replication on each strand, you must also know the directionality of DNA that creates the need for these two different processes. And as I've explained previously, each DNA strand runs in the opposite direction of the other. As one is read in the 5' to 3' direction, the other one would be read 3' to 5', according to the manner in which the positions of the deoxyribose sugar is highlighted. And you must know that DNA polymerase only works in the 5' to 3' direction. And if DNA polymerase didn't recognize the directionality of the two DNA strands or work in the direction that it works, nothing in replication would be reproducible and life could not exist. You must also know which base binds to which other, like as we explained, G with C and A with T, and how DNA polymerase can recognize and utilize this binding pattern. And you must know which nucleotides are used for DNA and which are used for RNA. So DNA polymerase only inserts the DNA nucleotides into DNA. And in this slide, we're looking at a DNA nucleotide without an oxygen, as the arrow is pointing out, at the 2' position, or a deoxyribose used in this nucleotide. And here's an RNA nucleotide with an oxygen at the 2' position, containing a ribose sugar. And as I explained in previous videos, RNA nucleotides and DNA nucleotides are different and will only work on their respective molecules. And you must also know all the other enzymes and RNA molecules required for each process and how and where each one will recognize and function in these processes. And you must know all these things and much, much more ahead of time in order to plan to strategically, intentionally, and intelligently use them in the design and creation of the two processes used in replication. Randomness, which is the basis for evolution, does not and could not create such patterns of these processes in replication. Randomness, by definition, is the opposite of organization, intelligence, logic, order, choreography, and patterns. Randomness does not result from intelligence and prescience because it's random. Randomness is like repeatedly rolling a dice, or dice is plural, right? Rolling dice. Or repeatedly selecting a different card from a deck of playing cards. You never know what you'll get. It always varies, and there's no intelligence, logic, or pattern to it. It's like telling a computer programmer to create a program by randomly selecting the figures that he or she uses. If a programmer just randomly entered figures at their whim, you'd never get a functional product. Randomness would result in a complete disarray of the components used in the two mechanisms of replication. Such a disarray would completely destroy their abilities to perform these essential functions. The fallout from this would be that their functions would be lost and replication itself would be lost. And the fallout from all of that is that life would be lost. Life would not be possible. Therefore, randomness, which is the basis of evolution, does not have the ability to create such an organized, intelligent, logical, ordered, and essential pattern of the mechanisms used in the process of replication. And in order for all of this to work properly, none of it can be left to chance. Therefore, as I've already explained, all of these molecules have to be here at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed from day one, which means they simply could not arise via evolution. And what's even more amazing about replication is how all of the information needed to make it happen comes from DNA itself. And speaking of coming from DNA itself, let me add something else here. Mitochondrial DNA is the additional DNA that exists in the mitochondria of our eukaryotic cells. Mitochondrial DNA is distinct from our genomic DNA. It's a circular molecule that contains much less DNA than our genome does. In humans, for example, mitochondrial DNA only spans about 16,000 base pairs. It's a little more than that. In humans, there can be from 1 to several thousand copies of these mitochondrial DNA in a given cell. Now, mitochondrial DNA encodes for proteins. In humans, there's 13, and RNA molecules. And again, in humans, there's 22 tRNA molecules and 2 that encode the small and large subunits of ribosomal RNA. These molecules are used to convert the chemical energy from our food into a form that we can use in our cells, namely adenosine triphosphate, or ATP. Because mitochondrial DNA is separate from genomic DNA, and because the ribosomes that mitochondrial DNA utilizes are similar to those of bacteria, evolution has claimed that mitochondria and mitochondrial DNA came from bacteria during evolution. This is what they call the endosymbiotic theory. Now, I already explained the problems with this so-called theory, so I won't I won't rehash any of that here. But what I do want to mention here relates to DNA polymerase. There are different families of DNA polymerase enzymes. The DNA polymerase that replicates mitochondrial DNA is called DNA polymerase gamma. DNA polymerase gamma is an enzyme that's composed of 3 subunits: a catalytic subunit which is encoded by the POLG1 gene, and a dimeric subunit encoded by the POLG2 gene. The DNA for POLG1 has been mapped to human chromosome 15, while the DNA for the POLG2 gene has been mapped to human chromosome 17. So, do you detect a problem here for evolution? If the mitochondrial DNA came from bacteria, why are the genes for the DNA polymerase used to replicate the mitochondrial DNA found within the human genome? So what you have to understand here is that the DNA polymerase used to replicate the mitochondrial DNA, DNA polymerase gamma, is different from the DNA polymerase used to replicate genomic DNA, such as DNA polymerase alpha, delta, and epsilon, and different still from the DNA polymerase used to replicate bacterial DNA, namely DNA polymerase 3. If mitochondrial DNA came from bacteria, the DNA polymerase used to replicate it would be the same as that of the bacteria. Getting mitochondrial DNA from bacteria without the DNA to replicate it is useless. So, what was the origin of the genes that encode DNA polymerase gamma in the human genome? How could the human genome know to account for the need for the other DNA polymerase before having the mitochondrial DNA? How could that have happened randomly? Why would genes necessary for the replication of mitochondrial DNA, an absolute requirement for the energy needed for our cells, and therefore an absolute requirement for life, be housed elsewhere if it all supposedly came from bacteria? Moreover, the large number of genes that encode the structural proteins of mitochondria are also found in the human genome DNA, as are the genes for the mitochondrial DNA polymerase. So once again, we have the same problem that I already pointed out with the mitochondrial DNA polymerase genes. How could and why would an engulfed bacterium insert all these mitochondrial structures and mitochondrial DNA polymerase genes into multiple locations across the human genome and yet keep the mitochondrial DNA separate and intact? Do you see the number of problems here for evolution? And I also have to point out that no bacteria have mitochondria or mitochondrial DNA. Bacteria do not have or utilize mitochondria, its DNA, or the genes it contains. They utilize a different mechanism for energy generation. But supposedly, in the past, some did. So where's the proof to support this claim? It is simply not observed in life, and therefore it makes no sense. And in addition, mitochondria are almost always received via maternal inheritance because they're in the cytoplasm of the egg cell. And let me point out here that there are examples of paternal inheritance in some mussels and some insects like fruit flies and honeybees and cicadas, as well as there are rare instances of this in mice, sheep, and cloned cattle. So if mitochondria and mitochondrial DNA primarily come from the mother, and they were supposedly engulfed by a eukaryotic cell at some time long ago, how would the first life form then divide its heritability in this manner? For example, if the first life form that engulfed this bacterium reproduced asexually, how is it then distributed in this manner? And if the first life form that engulfed this bacterium reproduced sexually, how did the male survive without it? I mentioned ATP a minute ago as this energy molecule for our storage of energy in our cells. Well, one thing that I didn't discuss is where the energy comes from that empowers proteins like DNA polymerase during replication and proteins like RNA polymerase II during transcription. And the answer is that the energy is found in the phosphate bonds of nucleotides like ATP. Now, the backbone of DNA, or RNA for that matter, only contains one phosphate group between each 2' deoxyribose, or ribose for RNA. Like this deoxy-ATP that we're looking at here. One phosphate group, right? And again, here we're focusing on a DNA nucleotide. So, see that no O down in there, right? Okay. But a free nucleotide, like this deoxy-ATP, can contain 3 phosphate groups. That's because when a nucleotide is incorporated into the DNA, or RNA, in that process, DNA polymerase for replication and RNA polymerase II for transcription remove the last 2 phosphate groups from the nucleotide before adding it to the growing strand. So they would remove these 2 phosphate groups right there, breaking these bonds in here between the phosphate groups, which are called phosphodiester bonds, provides the energy necessary to perform these functions. Therefore, you must know the energy requirements of DNA polymerase and RNA polymerase II, and the fact that the high energy phosphate bonds can supply their energy demand ahead of time in order to plan to strategically, intentionally, and intelligently use these phosphate bonds exactly as required in the structure and essential functions of each DNA nucleotide, RNA nucleotide, DNA as a whole, RNA as a whole, DNA polymerase, and RNA polymerase II. And let me again say, the molecules that I mentioned during the discussion of replication are all part of the 300 or so molecules I've discussed many times already that are needed for the life-essential processes of molecular biology. For example, the topoisomerase, gyrase proteins, which are those guys there, right, are essential to relax the extensive supercoiling of DNA introduced by helicase, which was that guy right there. However, helicase is also necessary to both unwind and separate the 2 strands of DNA ahead of DNA synthesis. If helicase was left unabated, in other words, without topoisomerase or gyrase proteins doing their job, this tension would eventually grind DNA replication to a halt. But without helicase, you cannot have replication because you couldn't unwind and separate the 2 strands of DNA. Therefore, you must know the requirements and specific functions of all these proteins involved in replication, as well as the structure of DNA and how they interact with DNA ahead of time in order to plan to strategically, intentionally, and intelligently create the structure of DNA and the structure and functions of these proteins exactly as required in the design of the essential DNA function of replication. But it doesn't end with just these proteins. DNA polymerase, which I'm showing there and there, right, must be able to bind DNA. Therefore, it needs the appropriate protein structure to do this. And DNA polymerase cannot initiate the synthesis of DNA without a primer. So it relies on primase, this guy right here. It relies on primase to provide a primer that it can use to initiate DNA synthesis. It must know the difference between the nucleotides used for DNA and the ones used for RNA, as I explained previously, and utilize only the appropriate ones for DNA. Without all of this, there is no DNA replication, and without that, as I said previously, life ceases. You should be able to see that all of these molecules are critically important for these processes and therefore critically important for life. With any of these molecules missing, critical parts of these processes won't happen. And if any of these steps in these processes don't happen, life cannot happen. Therefore, as I've explained, all of these molecules have to be here at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed from day one, which means they simply could not arise via evolution. And as I've reviewed in previous videos, this is once again an example of irreducible complexity to the utmost. And I'm going to end this video here, but come on back for the next one. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching. And as always, we'll leave the light on for you.